Hi everyone and welcome to Miss Estruch Biology, and in this video we're going through topic 16, one of my favourites, which is inheritance. So make sure if you do want any extra help fully understanding all the theory, knowing the key marking points that come up and key examiners tips, then check out my A-level notes, which I've linked in the description below. But for now, let's get into my favourite topic, inheritance. Topic 16, which is looking at the passage of information from parents to offspring, or in other words, inheritance. And we're going to start by looking at these key terms for inheritance. So haploid, also represented by the letter N, means that you have one set of chromosomes in those cells. Diploid cells, or 2N, means you have two sets of chromosomes. And homologous pairs of chromosomes are two chromosomes that contain the same genes in the same position. Now we're going to look at meiosis first of all, and meiosis is a type of cell division. It's known as reduction division because it results in the production of gametes, meaning you end up with half of the number of chromosomes compared to the parental cell. This is crucial because when two gametes fuse together during fertilization, the resulting zygote will then have the correct diploid chromosome number. And without that reduction division, the chromosome number would double every single generation when fertilization happened, leading to an exponential increase in chromosomes and the number of genetic material. Now, unlike in mitosis, there are two nuclear divisions. That's why we end up with reduction in the chromosome number. And we get four genetically different haploid cells. So how that happens then, as we said, we've got two nuclear divisions. We get four genetically different haploid cells. We have two rounds of division that are referred to meiosis 1 for the first round and meiosis 2 for the second round. Both stages include the same stages: prophase, metaphase, anaphase, telophase, and then cytokinesis to split the cytoplasm and the cell membrane to create two new cells. And here's just a reminder of these two key terms, haploid and diploid, because that's going to come up as we go through the stages of meiosis. Now, the key stages that you need to know about actually happen in meiosis 1. So the way we get these genetically different haploid cells, the genetic difference is introduced by independent assortment of homologous chromosomes and crossing over, both of which happen in meiosis 1. So crossing over, this happens in meiosis 1 when the homologous chromosomes pair up at the equator. And when they pair up at the equator opposite each other, we call that a bivalent. So this here would be a bivalent because it's two homologous chromosomes aligned next to each other. What can happen, not all the time, but sometimes when that bivalent forms, the non-sister chromatids, which means the chromatid from the two different chromosomes, can cross over when they're in that bivalent. And the exact position where they cross over is known as a chiasma. Where those non-sister chromatids cross over, it can put tension on that chromatid, causing parts of the chromatids to break and then swap, and they're exchanged. And because they've now swapped a section of that chromatid, they have exactly the same genes because they're homologous chromosomes, but they might have had different versions or different alleles of those genes. And that results then in new combinations of alleles in the resulting gametes that will be made from these chromosomes. Independent assortment also happens in meiosis 1, and it's in metaphase 1 when the homologous pairs are lining up at the equator to make those bivalents. But this time the concept is it's random which side of the equator the homologous pairs align. So whether you have the maternal or the paternal on the left or the right for all of those homologous pairs. So we can see here we've got one option where you've got all of the blue on one side, all of the red, but we've got a different combination in this option and a different combination here and a different combination there. And because it's random which side of the equator they go, you're going to end up with different combinations of maternal and paternal chromosomes in the gamete, which introduces genetic variation. And you can actually calculate the number of possible combinations you could get of how those homologous pairs could align, and therefore how many different possible gametes you could create by using the formula 2 to the power of n, where n is the number of homologous pairs. And it's 2 because we have 2 chromosomes in that pair. So for humans, we have 23 homologous pairs. So to work out how many possible different gametes we could create because of independent assortment of homologous chromosomes, it'd be 2 to the power of 23, which gives us over 8 million different combinations. Next then, we go on to the roles of genes in determining the phenotype. Here's a selection of essential key terms you need to know for inheritance. So we've got genotype and phenotype, the genetic constitution of an organism, so it's the alleles it has for a gene, whereas the phenotype is the expression of the genes and its interaction with the environment. Homozygous means a pair of homologous chromosomes carrying the same alleles for a single gene, whereas heterozygous is when you have 2 different alleles for a single gene. We've got our recessive, dominant and co-dominant alleles. Recessive alleles are only expressed if there are no dominant ones present. Dominant alleles are always expressed. And co-dominance is when you have 2 alleles that are equally dominant, and therefore if you have a copy of both in your genotype, both will be expressed in the phenotype. Multiple alleles is when there is more than 2 possible alleles for a particular gene. Sex linkage is when a gene is located on the X chromosome. Autosomal linkage is not about the sex chromosomes, but it's when genes are located on the same chromosome, but not a sex chromosome. Epistasis is when one gene modifies or masks the expression of another gene. Monohybrid inheritance is when we look at the inheritance of one particular gene, and dihybrid inheritance is when we look at genetic crosses for 2 particular genes. So these would be good to turn into flashcards to test yourself and to learn. Now, you commonly have to do genetic diagrams to determine the probability of different either genotypes or phenotypes in the offspring based on parental genotypes or phenotypes you're given. And we use different coding to represent the alleles. So this is just a summary of the accepted coding. In monohybrid inheritance, which is what you would have done at GCSE, when you're looking at the inheritance of either a dominant or recessive allele for one particular gene, we pick one letter, and that represents the gene, and you have the capital letter, which represents the dominant allele, lowercase letter represents the recessive allele. In co-dominance, because both alleles are dominant, it wouldn't work to use one letter that were capital or lowercase, because they'd both have to be capital and therefore it would look the same. So that's why we changed the coding system, and we use a base letter that represents the gene, and then we do a superscript letter which represents the allele. And we use 2 different letters, both capitals, to represent the 2 different co-dominant alleles. We have to do the same thing in multiple alleles because you have more than 2 alleles for a gene. Therefore, we can't just do capital and lowercase with the same letter. So that's why here we can see we've got the base letter for the gene and we've got the 3 possible alleles for blood groups shown there. Sex linkage, we do the base letter showing the chromosome and then the allele goes superscript. And you don't get the sex-linked genes on the Y chromosome. So you never have an allele written next to the Y. You only have an allele written next to the X. Autosomal linkage, this is just using the same system as the monohybrid, but that will always be a dihybrid example. So that's the inheritance of 2 genes. That's why we've got 2 different letters, but it should be capital or lowercase for dominant and recessive. Same with epistasis. That's always a dihybrid example because one gene can mask the expression of the other gene, but you'd pick one letter for one of the genes, another letter for the other, capital and lowercase to represent dominance and recessive. So let's go through some genetic cross examples to look at how this all works. Starting with monohybrid. This one we've got cystic fibrosis is caused by a recessive allele, and we've got if 2 carriers reproduce, what is the probability that they'll have a child with cystic fibrosis? So here we have the parental genotypes. They're both carriers, so they need a capital and a lowercase, a dominant and a recessive. I've then done my Punnett square, showing the possible gametes for both parents, drawing those gametes together to show the 4 possible genotypes of the offspring. And then I've put in what the genotype for someone who is a cystic fibrosis sufferer must be if it is caused by a recessive allele. So from that, we can see we have 25% probability of a child having cystic fibrosis. If they then asked what is the probability of it being a girl, which they did here, we then need to times by a half, because it's always 50% probability of having a boy, 50% probability of having a girl, or male or female. Then if we have a look at this codominance example, cows can be red, white or roan. We call this colour red when we're describing cows. Red and white are both dominant, and you only get roan, which is this mixture of white and red, if an individual has the codominant genotype. So they have a red and a white allele, so both are expressed in the phenotype. And in this example, we're told that we have 2 roan cows reproducing, and we're asked what is the probability they'll produce red offspring. So here are the parents' genotypes. If they're roan, they must have one of each allele, a red one and a white one. We've then got our Punnett square showing the parental gametes on the outside, just as a single letter, because you only have one allele for each gene in a gamete. Joining together those gametes to show the 4 possible genotypes, and I've written in the box the 4 matching phenotypes. And from that we can see we have 25% probability of having a red cow with those parents. Blood groups is an example of codominance and multiple alleles. There are 3 alleles, blood group, the allele for blood group A, an allele for blood group B, an allele for blood group O. But A and B are both dominant. So if an individual has A and B, they'll have the 4th phenotype, which is blood group AB. Blood group O, or the allele that codes for blood group O, is a recessive allele. So you're only blood group O if you have 2 copies of that recessive allele. And that's why you could have allele B and allele O and still be blood group O, because B is dominant over O. And the same with blood group A. Allele IA is dominant over allele IO. So here's our potential genotypes. There's our potential phenotypes. And in this example, we've got parents with blood group AB and blood group O reproduce. IA and IB are codominant and IO is recessive. What's the probability they'll have an offspring with blood group A? So first of all, we need to work out the parents' genotypes. And if they're blood group AB, one parent must be IA-IB, and blood group O, one parent must be IO-IO. So then if we split it up to see the gametes that both parents would be able to produce, we can then do our Punnett square to see the 4 possible genotypes. And then in the box, I've also written the corresponding phenotype. And from that, we can see we've got 50% probability of blood group A. We're now moving on to sex linkage. We're going to look at the example of colour blindness. Colour blindness is caused by a recessive allele found only on the X chromosome. And we've got the example of if a non-colourblind male reproduces with a female carrier of the allele, what's the probability of their children being colourblind? So the female is a carrier, which means they must have 2 different alleles, so the capital and the lowercase. And recessive is colour blindness, so that is our colour blindness allele. And they're reproducing with a male, so they are XY. And the male does not have colour blindness. So the males only have one version of the gene, and the version they have is the capital R for non-colourblind. So if we have a look at the Punnett square, we can see the gametes on the outside, the 4 possible genotypes, and the matching phenotypes. So this would be a biological female with colour vision, because it's a dominant allele, biological female with colour vision, and that'd be a colourblind biological male, missed off the biological male there, and then the biological male with colour vision. So the probability of having a child being colourblind is 25%. Next is epistasis, and that was the definition we said is where one gene influences the expression of another gene. An example of this is coat colour in mice or coat colour in Labradors. Fruit colour in vegetables is another one. So colour is quite a common example. So if we go through the coat colour in Labradors example, one of the genes controls whether pigment will be expressed, so produced. The dominant allele for that gene means the pigment will be made. The recessive allele codes for no production of pigment, and in which case for a Labrador, if they have 2 copies of this recessive allele, it will mask the expression of the second allele, and they will always be yellow in colour, or golden, whatever you want to call it. Gene 2 is the gene responsible for what colour the pigment will be. So capital B would be a dominant allele, and that one codes for black fur. Recessive allele is for brown fur. So here's our 3 possible phenotypes, but gene 1 can mask the expression of gene 2. Because if an individual is lowercase e, meaning 2 recessive alleles, it doesn't matter what combination of alleles they have for gene 2 is not going to be expressed. So if we go through an example, here we have 2 parents heterozygous for both genes, and because they've got at least one capital E, that means the pigment will be expressed, and because they've got a capital B, that means the pigment is black. So this would be 2 black Labradors. The possible gametes that they could both produce would be all of the options of the capital E, lowercase e, capital B, lowercase b in your gametes, because you only have one copy of each gene in each gamete. So here's our 4 possible gametes for both parents. That then gives us this Punnett square. It's a large one because there's 4 gametes for all of them. And we can then put in what the possible genotypes would be and the possible phenotypes. So we can see here any that have a capital E are going to be either black or brown. Any that have 2 recessive E alleles will always be yellow, and it masks the expression of that second gene. So in this instance, we've got a 9:4:3 ratio of those phenotypes. So a dihybrid cross is where the inheritance of 2 genes is considered at the same time, which we actually just saw there in that epistasis example. A common example linked to this is Mendel and his peas. So he looked at pea plants, considering the gene for colour and the gene for whether it was round or wrinkled. Only he didn't know genes at that point. He was just talking or noting down the colour and texture. And when we do genetic crosses, we should always show and label where possible the parental phenotype, parental genotype, possible gametes, the offspring genotype, correctly match up the phenotype to those genotypes and give the proportion of each phenotype. And that's why often in the exam, they give you this space to fill in each of those criteria. So we've got a round yellow and a wrinkled green. And in this example, we are being told that the round yellow is homozygous dominant for both of those genes. Wrinkled green would have to be homozygous recessive for both, because wrinkled and green are recessive. Now, you aren't expected to know that. That's information you would have had to have been told. Then we work out the possible gametes. And in a dihybrid example, every gamete should have 1 copy of each letter. So 1 of the R's, 1 of the Y's, to indicate 1 copy of each gene. Now for this parent, they can only produce gametes with a capital R, capital Y, which is the 2 dominant alleles. And this parent can only produce gametes with the 2 recessive, so recessive R, recessive Y. We can then do a Punnett square. But actually for this one, we don't need to do a Punnett square, because if this parent can only donate this gamete, this parent can only donate that one. If we join them together, every single option on the Punnett square would come out with this heterozygous offspring genotype. So 100% would have that genotype, which would mean 100% would be yellow and round, but heterozygous. So you can actually cross those together now. This commonly is shown. If you were then to cross F1, which is the first generation, which is what we just created together, we've now got 2 parents who are both heterozygous for both genes. For this, we now have these 4 possible gametes for both parents. And now we're going to end up with one of these large Punnett squares again, where we'd have to put in all of the possible genotypes, combining those gametes. And then we can put in all of those possible corresponding phenotypes, and then we can work out the ratio or proportion of the phenotypes. and we have 9 yellow and round, 3 green and round, 3 yellow and wrinkled, and 1 green and wrinkled. So a 9-3-3-1 ratio. And in any dihybrid cross, if you are given a scenario where both parents are heterozygous for both genes, you will always get a 9-3-3-1 ratio. Unless there is autosomal linkage, which we're going to have a look at, or if there's crossing over in meiosis, or if epistasis has occurred as well. So crossing over in meiosis, that creates new combinations of alleles in the gametes. So that means what you predicted in your Punnett square may not happen. And this links back to what we said right at the start of the video. Crossing over introduces new possible combinations of alleles in the gametes, and therefore we don't get what we expected from our Punnett square. I'm going to come back to that concept once we've gone through autosomal linkage. And we said autosomal linkage is when 2 different genes are on the same non-sex chromosome. So we can see here a homologous pair of chromosomes, gene 1 and gene 2 are on the same chromosome. So they are linked autosomally. We're going to apply the example we've just done. So gene 1 is for whether it's yellow or green. Gene 2 is for whether it's wrinkled or smooth. And we said that the parents were both heterozygous for both genes, which we can see. This is the homologous pair of chromosomes for one of the parents, and they've got the capital on the recessive, the capital on the recessive. It just so happens to be, because they're linked, the 2 dominants, so the 2 capitals, are on this chromosome of the homologous pair, and the 2 recessive alleles are on the other chromosome in the homologous pair. And that means we can't actually make 4 possible gametes now. We can only make 2 possible gametes, because in meiosis, when these homologous pairs are separated, that will mean this will go on to make 1 gamete, which will have to have the capital R and the capital Y, and this chromosome will go on to make another gamete, which means that one will always have lowercase r and lowercase y. And therefore, these are the only 2 gametes that this parent can make, rather than the 4 possible ones that we looked at initially. So here were the 4 possible ones that we said initially if there was no autosomal linkage. But because there is autosomal linkage, these are the only gametes that the parent can produce. And if we have 2 parents who are the same, reproducing with those 2 possible gametes, these are the 4 possible genotypes we get, and the corresponding phenotypes. So our ratio is now 3 yellow and round to 1 green and wrinkled. So with 2 heterozygous parents, you would expect to get a 9-3-3-1 ratio, unless there's autosomal linkage, when you could get a different ratio. And for this particular example, we got a 3-1 ratio. Now, we did also say unless there's crossing over that happens. So sometimes you might get an autosomal linkage exam question where you're led through all the information we've just gone through to work out you'd expect a 3-1 ratio. But they then might say, however, scientists did this cross, and what they actually observed was these phenotypes in this proportion. And you could be asked to explain their results. And it's only possible to get those 2 extra phenotypes if crossing over occurred in meiosis between these homologous pairs to create new combinations of gametes. So here we can see those linked genes. These were the 2 recessive ones on one of the homologous pairs. Here were the 2 dominant ones on one of the homologous pairs. They now look like double structures because this is showing after DNA replication. And in meiosis 1, when they line up at the equator to form a bivalent, we can see the non-sister chromatids crossing over to form a chiasma, break and swap, and that then means when they do then separate in meiosis 1 and meiosis 2, ultimately we get 4 gametes which have new combinations of alleles in the gametes. So to explain these results then, we'd have to say yellow and round and wrinkled and green are the most commonly observed because those are the genotypes that are a result of fusing the capital R and Y and the lowercase r and Y together. And those were the ones that are Most common because they were not formed by the crossing over, and crossing over is actually quite rare. Now, out of the yellow round and green wrinkled, we've got fewer green and wrinkled because that's the recessive combination. So you'd have to have 2 copies of the R's, 2 copies of the lowercase y's to be green and wrinkled. So that's why there's fewer green and wrinkled compared to yellow and rounds. Then why we don't have very many at all of the middle 2, those 2 are a result of crossing over to create the capital R lowercase y and the lowercase r capital Y gametes. And crossing over doesn't happen all the time, it's rare. And therefore we wouldn't get very many of these 2 genotypes, which is how we'd get those 2 phenotypes. So that would be your explanation of those results, combining the concept of autosomal linkage and crossing over. Now, throughout that, I was talking about what we expected to see versus what we actually observed. And that leads us into using the statistic of chi-squared in inheritance. So chi-squared is the statistic to investigate differences between frequencies, and it can be used to determine, is there a significant difference between the frequency you expect and the frequency you observe? And in inheritance, that links to what we expect from the Punnett square, and we can compare that to what we actually observe if you count literally the phenotypes that are there. And here's an example of this. Corn can be purple or yellow or smooth and wrinkled in terms of colour and texture. Using the results of the Punnett square and then counting the number of actual corn kernels of particular colours and textures, you can look at whether the frequency you expected is significantly different to the frequency you observed. So we're told here that purple is dominant, yellow is recessive, 2 heterozygous parents were crossed. This is just dihybrid, simple genetics. There's no autosomal linkage. And the expected ratio is 3 purple to 1 yellow. So that is our expected ratio. And we're told that a student then counted them and they had 21 purple kernels and 13 yellow kernels. Does this follow the expected ratio? So what we would then need to do is calculate the chi-squared, but I'm actually jumping straight to the result here so you can see how we'd use this as a conclusion. So our null hypothesis would be, for this example, there's no significant difference between the expected and observed frequency of the colour of the corn kernels. The chi-squared value was then calculated to be this. We've got 1 degree of freedom because there were 2 categories. So if we read across to 1 degree of freedom at p equals 0.05, that means our chi-squared value is lower than the critical value. And therefore we have more than 5% probability that the difference between the observed and the expected is due to chance. And therefore we have to accept the null hypothesis, meaning there is no significant difference between what we observed and what we expected. So in other words, the frequency of the corn kernels that we observed does match what we expected from our Punnett square, and it confirms that Mendelian genetics ratio of 3 to 1. Test cross is the final thing we're going to look at, and these are used to determine the genotype of an individual expressing a dominant phenotype by crossing it with a homozygous recessive individual. And this helps in revealing whether the dominant phenotype an individual has is determined by a genotype that's heterozygous or homozygous dominant. Because if you have the dominant phenotype, that could be someone who's homozygous dominant or heterozygous. So if we use an example of Mendelian genetics or inheritance, specifically the inheritance of flower colours in pea plants, where purple is dominant and white is recessive, we could perform a test cross to determine the genotype of a purple flower. So what we would need to do is cross the purple flower plant that we're trying to identify the genotype of with a white plant, because we know that white plant has to be homozygous recessive. From the results of the Punnett square or the offspring breeding together, we can then work out whether the parent was homozygous dominant or whether they were heterozygous. Because in this Punnett square we've crossed the purple flower with a white flower. And because we do have a white flower here, that would only be possible if the purple parent was heterozygous because to get a white flower, both parents have to have a white allele. So in these test crosses, if you get any offspring that have the recessive phenotype, it confirms the parent that was the dominant trait must be heterozygous. And that's what we've just got here explained. So you can pause, read through that bit, but it's what I was just talking through on that last slide. So that takes us to the end of this topic. Hope you found it helpful. If you did, don't forget to hit that subscribe button so you don't miss out on any of the latest videos.